I'm Ms. Hanifa, and I'll be speaking on behalf of the Human Cell Atlas Developmental Biological Network, as well as representing my co-coordinators, Deanne Taylor and Stanley Narson. Please do not hesitate to get in touch if you're interested in our network, and the email is as below, development@humancellatlas.org. We first came together as a biological network with this great ambition of organizing the Developmental and Pediatric Cell Atlas meeting in 2020. Unfortunately, the coronavirus pandemic hit, and so we changed our idea of a meeting into a seminar series. And you can see here the list of the topics that were covered during the seminar. And also in the 2 time zones. And if you're interested, please go and visit this website for the schedule and past recordings. In addition to that, we also came together as a community to write a roadmap for the Human Developmental Cell Atlas. And this was a massive effort from many individuals across many countries. And in doing so, we identified several things which I'd like to share with you today. Firstly, specific challenges relating to studying human development. As you can see here, from the zygote all the way to the fully formed fetus, there are a lot of changes in morphology and also transient structures and all of these things happening across space, anatomical space and gestational time. So this is really quite difficult to capture in its entirety, particularly very early stages during development, where access to embryonic or fetal material is very difficult. And we therefore realized that for some particular gestational stages, we may need to rely on in vitro culture systems, organoid models, and animal models. And this will function to fill in the gaps in certain situations, but also provide us with a way of functionally validating our findings. And in doing so, we will interface very well with other networks, such as the Organoid network and also the developmental biology community at large. So how would you build a developmental cell atlas? And throughout this meeting and with the Human Cell Atlas type of work, you'll realize that this involves multidisciplinary teams using multiple different types of technologies and high cutting edge data analysis and bioinformatics techniques. So I will illustrate with how most of the Human Cell Atlas profiling is currently being done, which is organ-based, and particularly for embryonic or fetal tissues, you can actually get the whole organ to analyze. And this involves both dissociating the tissue to extract the cells for analysis and also combining it with spatial in situ analysis. So these are the sort of technologies that you can employ to study the development of the heart across several gestational stages and integrating all of that information together to reconstruct an atlas of the developing heart, as illustrated in this publication below. These are the publications that we have generated throughout from the Developmental Cell Atlas initiative, and all of the manuscripts are actually part of the Human Cell Atlas publications. And you can see that the vast majority of these have involved analyzing single organs, but also some of these manuscripts have involved 1 or 3 organs, but here are several examples where it's a whole fetus or a whole embryo where several organs from one embryo or fetus has been used for the analysis. In addition to this wealth of data, we have, the community has also provided these data in browsable formats through web portals, and you can see here the various web portals that are available for you to interface and intuitively browse the data. We heard yesterday how we query the atlas, and the consensus was the initial query is going to be about cellular molecular processes, and in this case, underpinning human development. And what do we expect from this type of query? And this is really with regards to the clinical relevance of such a reference atlas. And I'm going to touch on three important areas where this is going to be clinically relevant: regenerative medicine and tissue engineering in the first example, and in several other examples, relevance to disease and therapeutic strategy. So with regards to regenerative medicine, you can see here organs from the human endodermal derivatives that have been, made into organoids from iPS-derived cells, and this allows us a good way to look against the reference data, but also with regards to how faithfully these organoid models recapitulate in vivo tissues. And certainly in some of the work that I've been involved in with several collaborators listed here, looking at the development of the blood and immune system across several organs and gestational period, you can see the types of blood and immune cells that are formed in the fetal liver, in contrast to the fetal bone marrow, where neutrophils or granulocytes first emerge. And you can now put these data sets together across organs to understand how blood and immune cells are developed across organs and lifespan, and also to use the healthy reference data set to understand a disease or perturbed situations. And here trisomy 21 causing Down syndrome, how this compares with disomic bone marrow, and also how you can use this data set to map infant and childhood leukemias. And certainly the idea of childhood cancers having a lot of conserved developmental programs has been shown in many publications, including for childhood cerebellar tumors and Wilms tumor. More recently, we showed how some of these developmental gene programs are relevant in inflammatory disease, and here in inflammatory skin disease, and also here in inflammatory bowel disease. But more importantly, a lot of childhood disorders are developmental based. And here's one big developmental cell atlas project led by Alan Shadotal to map gonadal development across human gestation, with the idea that this reference data set is going to be helpful in understanding differences of sex development, which is particularly important. And lastly, just to show how with increasing numbers and increasing the time period of studying development and into pediatric cohorts, how we can begin to look at disease-causing variants and how that may affect developmental disorders, to have a better understanding of these diseases and to innovate new therapeutic strategies. And I'm going to finish now to give a shout out to the meeting that we never had in 2020, which will now happen in 2021 in a virtual setting. And if you are interested, please register to the meeting, which will be in August 25th to the 27th. And I will stop here, and I'm very happy to take any questions.
Thank you very much, Maz. Waiting to find out if there are questions coming in from the audience. There are no questions so far. Please, if you have a question, use the Q&A section in the main stage to enter your question and vote on other people's questions. I have a quick question. How will we think about ethics for the developmental atlas? Do we need to have a separate activity thinking about that, or should it be integrated into all the individual projects?
So far, we've relied on, most of the researchers have relied on repositories where there are existing ethics that have been taken into account. So one good example would be the Human Developmental Biology Resource, which is funded by Wellcome and MRC, that has been around for 20 years, based in both London and Newcastle in the UK. And here, the resource is specifically dedicated to providing embryonic and fetal material for researchers globally. And you can register your project, and if, with the kind of relevant ethics approval for your project, you can interface with HDBR to obtain and procure the samples that you need. And there are other sort of resources now, also in France, with Hugh Decker, the Human Developmental Cell Atlas, funded by INSERM. So that has been some of the ways that researchers have managed to undertake studies in human development with the auspices of appropriate ethics and relevant consent.
Great. So a question is coming in. Are there already examples of how HCA research and resources are translated into the clinic?
So I think a good example of that would be with regards to some of the insights from studying. I'll give an example of childhood cancer. I mean, I think you can see how they're going to be translated into clinic, but what is quite difficult to know is what we can't achieve within the time frame that we've had, really from HCA first beginning in 2017, is to actually have the kind of like end product in clinics. So I give some examples. So a lot of the resources for like the web portals, you can actually query the expression of disease-causing genes at single cell resolution. So one example from our liver and bone marrow data set is looking at a particular type of gene that causes a specific immunodeficiency. And, you know, this is where a researcher then actually found this and said, I can explain why my patients have, you know, impaired or have thrombocytopenia or impaired megakaryocyte differentiation, because it's a crucial gene in that differentiation pathway. So those are the type of insights that you can gain in terms of explaining phenotypes, clinical phenotypes that you see that is not explained with the current knowledge, and also in terms of what may be suitable therapeutic targets. And those resources are extremely helpful for clinicians in an intuitive web portal to look at, you know, where the genes of interest, and we've incorporated that certainly in the developmental atlas.ncl.ac.uk. Secondly, in terms of, I think, some examples of cancer, you can actually look at pathways that can be targeted to drive the differentiation, because some of these childhood cancers are where cells are suspended in a particular state and don't complete the differentiation. For example, with Wilms tumor, they don't quite form the nephron, or in some of the leukemias, they are suspended in a particular state that doesn't go through the full differentiation. And if you can actually target those, that can drive that. But all of this will take a few years to actually be translated for patient benefit, because you need a lot, you need clinical trials, etc., and they can't be achieved in the time scale that we've been operating.
And I suppose that's going to be a big focus of the developmental and pediatric atlas meeting. So if people are interested, there'll be a really great discussion at that meeting. Absolutely.
I mean, what we want to do is drive these things forward. So if we can engage with the community, for the pediatricians and, you know, also developmental biologists, I think this will just push through the translation to the clinic.
And I guess we should also mention that this, about an hour after the session, there's going to be breakout sessions for all the biological networks, and there'll be another good opportunity to discuss many of these topics. So there's a question from Dana Payer about, Maz, what do you feel are the biggest computational challenges for you right now?
Well, I guess for the developmental cell atlas, rather than me specifically, is how I would interpret the question. Thanks, Dana. A lot of it is, you know, in terms of what you get with development is you kind of get how things are developing. You know, your snapshot isn't just differentiated and isn't just the stem cells. You get the whole spectrum. But a lot of the models, for example, for me in inferring trajectories, just looking at one direction, and it doesn't take into account the complexity of how cells are moving across all of the different states. So we may be very narrow in terms of how we can actually understand, you know, the fate transition from, you know, a stem cell all the way to the differentiated progenies. That's one. The second is essentially, how do we, you know, integrate all of the different parameters that we are measuring together in a way that doesn't sort of like, you know, overcome or eliminate the biology, a bit like the saying, you know, throwing the baby with the bathwater, because you try to correct everything, but in that process of, you know, correcting for artifacts, you run the risk of losing biological information. And this is true for sort of studying across gestational stages, because you might not have enough samples per gestational stage, and that can be quite difficult. So that's another computational challenge, in my opinion. Yeah.
Great, thanks. And Bruce Arano asks that many embryonic and fetal cells seem to be precursors of more mature, more than their more mature counterparts, but are nevertheless different. And he's wondering what strategies are most practical and enabling for entering this information into the cell ontology.
This is a really good question for several reasons. When we annotate developmental cells, we basically infer from our knowledge of postnatal and adult cells. And therefore, to some extent, we pigeonhole them into the way that we have the framework for. And I think more needs to be done to actually pull out what's different about those cells, and therefore have a framework that allows us to kind of like retain that information, but without complicating ontology, because they are similar, but they're also different at the same time. So how do we keep this information and not lose it? That's a challenge. I don't know whether I've answered that.
Yeah, I think that was great. So thanks, Maz. So yeah, thank you very much for your talk. We're going to move on to the next talk, and I'd like to invite Sasha Mishran and Martijn Nouwen to the stage, who will be talking about building the lung cell atlas brick by brick.
Okay.
While it's getting set up, go ahead. While it's getting set up, I should just mention there were many other questions in the Q&A, and so we can look at those and answer them. So, sorry, Martijn, you can start now. Thanks.
Thanks. So, sharing screen. So hopefully you can all see this. So thanks for the invite to present the lung cell atlas also here. And the first and last thing I should stress, and I really want to stress, is that we're a very open and inclusive community, of course, like all of HCA. And please, if you think that the lung corner of the HCA could be, the place where you feel at home, join us. You're more than welcome. And HCA Lung has been going at it already for quite a while. And we're really sort of an allegiance of all independent groups, like most of the biological networks will be, each making their own contribution while also trying to coordinate within the network, what are critical steps that need to be taken in a coordinated fashion. Within this community, there's a few main consortia, such as the Chan Zuckerberg Seed Network, an HCA project, DiscoVer, and there's of course LungMap and HubMap, which are NIH-supported consortia. These are the larger consortia, but there's also a lot of groups that are outside of these consortia, but still making huge contributions to the network. So please feel free to join us. It's not, you really can make an effort and make a contribution to the network. So I thought it would be nice to, now that we're almost 5 years in, to actually revisit a little bit how the network came to be. And we started off in this kickoff meeting in October 2016 already, so almost 5 years ago in London, where there was no lung network. There was an epithelium working group or something similar. And we were sitting there with people from gut and from pancreas, and then also a few people from lung. And clearly, we rapidly decided that we needed to split up into the organ systems. So the lung really had its own first breakout during the October 2017 meeting at the Weizmann Institute. And then we published the white paper as the HCA overall and also our own HCA lung approach in 2018, 2019. And also around those times, the data papers started coming in, with the first one really already being there in 2016. So actually predating the HCA, if I may say so, and a lot of papers being produced. And this, I could fill the entire slide if I wanted to with papers from the HCA lung group around the single cell atlases of the lung. And then in 2020, COVID-19 hit us all, and clearly this had a huge impact. And for HCA Lung, this really forced us to collaborate and try to make an impact early on in the pandemic. And that is actually also what happened. And this last Nature Medicine paper that you can see here was just published a few months ago, because this really forced us to take all the data sets within HCA lung that are available, and then merge those and integrate those to really start answering questions and doing meta-analyses and looking into demographics, etc. So really also accelerating sort of a technological advance within the network. As you can see here, the HCA lung cell atlas is spread throughout the world, but it is slightly biased towards Western Europe and the US. So I would really want to invite people from Latin America, from Africa, from Asia, or Oceania to really join us and sign up on the website so that we know you're there. We know you want to contribute, and we'll definitely invite you into our activities. Most of it is still virtual anyway at this moment. And here's just a view of the network that contributed to this last Nature Medicine paper. And of course, you could also be part of these sort of efforts because we feel that really combining all the data sets is where the power is for the Human Cell Atlas. Now, if you just want to access the data, of course, that's also possible. And this has already been highlighted yesterday as well. So there's COVID-19 cellatlas.com is where a lot of the data sets you can just download, both healthy data set and diseased data sets. The single cell expression atlas, and there's actually a poster on this today, not on lung, unfortunately, but still from the same group, where you can really start to interact with the data and say, I want to look at this anatomical structure and then the cell types that sit there and what are their expression profiles, etc. So this is a more advanced sort of interaction with the data that you can have, a little bit beyond CellXGene, which is the go-to sort of platform that most of us use at this moment. And this is being developed at EBI and will also be implemented and become fully available for lung with the data sets that is out there. Of course, the consortia also have their own data infrastructure. So here's the HCA lung map interface where you can just look at the data sets that are in there. And this is being filled quite rapidly. And LungMap also has the ambition to become the one place where you can find all data sets regarding lung. And then on top of this, or in parallel to this, there's also this top cell platform at the Cincinnati Children's Hospital, where you can actually do analyses on these data sets. And you can see here's one of the data sets that I contributed to on top, but there's actually much more in there. And then if you do overall analysis, you can generate these sort of pictures, which says which genes are expressing what cell types, etc., in the different data sets together. So that's all very nice and worthwhile, I would say. And of course, we'll also all be filling the DCP within the HCA, but I thought that would be a little bit superfluous to put up here. Then one of the more recent advances within the network is this publication or this release of Azimuth from the from Raoul's lab, where where you can really project your own data set onto existing references to actually see what is the cell type annotation that other people gave to this cell type, and then integrate those to a certain extent. So for instance, here you can see, and the authors also really request that you upload your lung references, because the more data sets are in there, the better the system becomes, of course. And the references used at this moment is the one from the Krasno lab, which was published in Nature in 2020, which was a very highly detailed annotated data set. So really a very strong data set with regard to cell type annotations. And that, of course, makes perfect sense to then, project the other data sets onto that. But this is still very much projection, but it works lovely. So as you can see here, there's other data sets from the community. So the Haberman data set, the Medicine data set, and they are then projected onto this reference. And actually you can use this very well to integrate the data to the point where you can say these clusters overlap, and these cell types might actually need a single name rather than 20 different names, depending who you're asking. So this works lovely, but it's still sort of a projection, and I hope I don't upset Raoul with this, but in the TEIS lab, there's actually an effort to really integrate the data sets into a single embedding by benchmarking integration methods. So this will also be presented here at the HCA meeting this year. And then really making a single data set of them that you can just query as one data set and then filling it up with all the data sets that are available. So this particular data set will be released shortly. It contains about over 100 donors and is built basically on this experience we had in the COVID-19 effort, and contains more than 14 data sets, close to 600,000 cells. So the number of cells can still increase, but the number of subjects in there is actually very appealing, I think, and allows us to do all kinds of things like, look at the cell type references, identify rare cell types, identify cell state transitions, but also look at demographic variables. So what does age do? What does smoke exposure do? What does ethnicity do to your cell types? And when we do this, especially with ethnicity, we are clearly still undersampling. So this is really a concern actually for the community. So what are next steps? Well, I think we need to really start to broaden our scope a little bit, where we try to include much more different ethnicities and also a better balance across the age range, for instance. But there's also clear things about cell type labels that we need to resolve. So here, and I stole this slide actually from one of the presenters yesterday, who presented about the ontology, so David, and he didn't show this slide in his presentation, but it was in his deck. And this goes to show that everybody, even a well-known cell type as an AT1 alveolar epithelial cell, everybody gives it a different name. So it's not just the labeling that needs to be resolved, but also the structure in all these data sets together clearly identify cell types. And now we need to give the proper name and, to the proper cell type and integrate it with existing ontologies. So this is from David, but also Katie Borner from the HubMap is very active in this, and they use these anatomical structure cell type biomarker tables. And these are very interesting discussions. I join them sometimes when I have time. And we also need to integrate our atlases with these sort of efforts. And I think that's going to be very useful. So with that, we would like to use the last few minutes to give a short update on the progress in the field at the moment. And Sasha is going to take on from here.
Thank you, Martijn. Thanks to organizers for putting together this meeting and handling all the challenges related to remote presentations. I'd like to present a few examples of the recent progress in lung atlas development. This is by no means a comprehensive overview. There is a lot of work that happens on the background and that we are not aware of. And with this, I would like to thank all the groups that were willing to share the unpublished data here. So here's a great example from Purushottam Tata's lab at Duke, where he explored a very specific microanatomic niche, which was currently understudied and underrepresented in the existing atlases. And small airways is a really unique niche that plays an important role in the development of many pulmonary diseases, including COPD. That's a great example. Using combination of careful microdissection, a specific skill that his group developed, and combination of spatial methods and single-cell sequencing, Tata's lab discovered several epithelial and mesenchymal subtypes that are unique for this niche. And Martijn, if we go to the next slide. And here's another example from Kirsten Mayer's lab, where she also focused on another microanatomical niche that is actually largely restricted to larger airways. And here we are talking about submucosal glands. And again, here using combination of deep profiling across multiple anatomical locations within the human lung, she was able to, her group was able to assess specific composition of the submucosal glands and discover several novel epithelial and mesenchymal and immune cell types that are restricted to this niche, but also to get a better understanding of the distribution of the immune cell types within the human lung. And of course, we are moving next to the spatial methods. Spatial methods, as you heard, are gaining a lot of importance in the building of any organ atlases. And here we have a great example from Doug Shepard's lab, who developed novel microscopy technique, which can be applied to existing methods, such as SM-FISH or immunofluorescence microscopy. But these techniques allow us to scan large regions of the lung and generate really large volumes of the data, which are essential for truly reconstructing single-cell data sets at the organ scale level. And another example comes from Tushar Desai lab, which developed novel tissue clearing techniques. Again, this technique can be applied to existing imaging modalities, but what it allows is to overcome challenges that are associated with the high autofluorescence, which is really big imaging problem in the lung. And then we're moving on to work from European consortium. It's a combination of several labs, and you can recognize several leaders in the field, such as Christos Samalakis and Joachim Ludenberg. Here's an example of the targeted in situ sequencing using the panel of specific genes. And I think it's also a very nice example of interactions between pediatric and developmental atlases, and the same technology is also currently being applied to development of adult atlases. And then moving to another work from Christos's lab is a method called screenshot. Again, it's a combination of existing platform, Visium platform, and also high resolution profiling at the single cell level. And this work is currently being in use by several consortia, for example, CZI Lung Seed Network, and also by European Discover Consortium. Good example of collaboration and interactions between different groups. As Martijn said, those reference integrated atlases that were developed, they were used actually to inform the panel design for these methods. And of course, you know, we are not nearly done. And for discussion on spatial methods, we started really good discussion yesterday during the lung booth session, and I invite everyone to join us today during the breakout session to continue this discussion. This session will be led by Herbert Schiller, who is an existing member of HCA consortium, and a couple new members who recently joined us, Elizabeth Dong and Nick Bandwich. So please join us today if you want to continue discussion on spatial methods or challenges in the development of the lung cell atlases. And of course, you know, as we said, we need higher resolution for specific anatomical locations. And we saw examples how going to specific locations and applying appropriate tools and appropriate skills for these locations gives us really novel information. Of course, we need the integration between existing data sets and improving integration with the existing metadata, both about environmental exposure, but also clinically relevant metadata. We need more information about location, so whether it's where the samples are coming from, how these samples can be mapped back into existing atlases, and also whether we can predict location by looking at the transcriptomic profile or proteomic profile of the sample itself. So, and of course, you know, these atlases also currently expose areas where we can improve. This is a good example that we really oversampled ciliated cells, and we need to start sampling other locations. So, and we are looking forward for others to join our group and contribute to development of the lung atlas. Thank you.
Thank you, Martijn and Sasha. That was really great. So we have one question from Mike Pausen, which is a technical question asking, how do you account for the different properties of single cell versus single nucleus, or steady state versus nascent transcription?
I think it's a good question, not just for the lung atlas. There are several computational tools, and I think, you know, there are people who are better suited to answer these questions. We had great presentations from Fabian, from Peter Harchenko, from Raul Satij's lab. There are methods, right? And obviously differences in modalities, they present challenges, but I think since, for example, single nucleus RNA-seq has advantage of being able to sample specific locations that are not, or specific cell types that are not accessible for single cell RNA-seq, we have to use this tool. We have to use single nucleus RNA-seq, and then we have to find the ways to integrate this with single cell RNA-seq. It's a good challenge to have.
Okay, thank you. So we're going to have a panel session after the talks, and so we'll have additional time to answer questions from the audience. So please, audience, keep the questions coming in the Q&A. But right now we have to move on to our next talk, so thank you again. I'd like to introduce Christoph Bach and Barbara Treutlein, who will be telling us about enhancing the human cell atlas with organoids.
It's my great pleasure to introduce to you the Organoid Cell Atlas and HCA biological network focusing on organoids. Organoids are a bit different than the organ-centric networks because we are not mapping the primary cells coming from the body, but the recapitulation of these of development in cell culture. Organoids provide, therefore, a kind of can shine light at the dark spots of normal development where we have very little access to certain phases of development, but also to the early phases of diseases. And perhaps the most powerful thing of organoids is that we can perturb these human cells in the dish, and we can perform high throughput mechanistic dissection and disease modeling. For these reasons, organoids and the concept of an organoid cell atlas were already part of the initial white paper of the human cell atlas, initially spearheaded by Ernst Clevers within this initiative. And we really have these two major groups, the pluripotent stem cell derived organoids and the adult stem cell derived organoids. So the goal is that what we are doing on organoids has a reflection in what many of the other biological networks are doing on the primary tissues. And eventually, kind of one of the most important things will be to build an infrastructure, a computational interface that allows us to readily move back and forward between the readily perturbable cells in vitro and their corresponding in vivo counterparts, such that we can really get the biological relevance from the in vivo data, but the ability to modify, test, evaluate in the organoids in the dish. So for concrete examples of these concepts, I'm handing over to Barbara Teutlein, who is co-coordinating this biological network together with me and who will present some recent work in this field.
Thank you, Christoph. It's my great pleasure to actually introduce or present to you efforts from the organoid bionetwork. So, I mean, organoids, as Christoph said, can be generated in on two different routes. We can take somatic cells from a human individual, reprogram them into induced pluripotent stem cells, and then use those to generate these three-dimensional human tissues in vitro, which in this case model development of these organs. We can also use adult stem cells and grow three-dimensional human tissues from those. And in this case, we rather mimic homeostasis and regeneration of these tissues. And organoids are really exciting since they can be generated from any human individual, and by doing so, we have avatars of healthy and diseased human organ development. And so, how can organoids enhance the human cell atlas when we add organoid data to the primary cell atlases? Well, as also Mars pointed out, there are some time points that are really difficult to access during embryonic development of humans, and organoids provide a great opportunity here. We can also perform time course measurements for the same individual for the development of a given organ. We can genetically and environmentally perturb these tissues. We can do this in high throughput, doing high throughput screening. We can test hypotheses from developmental biology in these cultures, and so learn more about the mechanisms underlying the development of these organs. We can model disease, we can directly trace lineages, and finally we can we have a way to reverse engineer and better engineer human tissues that can then be used even for potentially cell and tissue-based therapies. And so simply, we think organoids are amazing. And of course, what I'm showing here, these 6 examples of organoid types are just a very small fraction of all the organoid protocols that have been developed in recent years. And I want to now, in the coming minutes, present to you on all these different points where organoids can really enhance the human cell atlas and provide examples from my lab and other labs in the field and other labs part of this network. So we can access difficult embryonic time points and perform time course measurements from the same human individual. And in this case, as a first example, I want to provide this data from our lab, where we profiled for the same individual brain organoid development from pluripotency up to 4 months of development. And you can then obtain these trajectories where you really profile the very early stages of human brain development, including formation of the neuroectoderm and the neuroepithelium that gets patterned, and then forms a diverse set of progenitor cells and radial glia that then give rise to different neurons in the different regions of the human brain. Of course, we are not limited to transcriptomics. We can generate multi-omic data, and in this case, for example, from RNA and ATAC derived gene regulatory networks. Regarding very early human development, one can make use of these gastroloids that have been developed, and in this case, exemplary data from Alfonso Martinez Arias and Alexander van Oudenarden's lab, where they analyzed human gastroloids in the first 96 hours of development, and for example, performing tomosynthesis on these organoids to analyze anterior-posterior patterning of these organoids. And this really gives you access to these very early phases of development that you otherwise can't really access. Now, as I said, we can perform perturbations both of genetic and environmental nature using these in vitro grown tissues. And as an example here, going back to this GRN, we can now actually annotate all these genes that we measure that are expressed during the development of these human brain organoids. We can assess what their relevance and function is by knocking them out, for example. So in this case, generating isogenic knockout line for this gene GLI3, as well as control cells that go through the same process, then growing organoids from them and performing single-cell RNA sequencing. In this case, we can clearly identify that this GLI3 gene is really essential to generate the human cortex, because when we have it knocked out, we really can't generate any cortical cells in these organoids, whereas the other brain regions are more or less normally generated in these organoids. We can generate, we can do these perturbations in higher throughput, of course. We can use methods such as PropSeq or Perturb-seq to knock out genes in higher throughput in mosaic organoids, where we would provide lentiviral guide RNA libraries to the iPS cells that are, for example, functionalized with an inducible Cas9. Then we can grow the organoids and use RNA-seq to phenotype the cells and to measure the presence of guides, which in these organoids, all kinds of different wild type and knockout genotypes are present. And then assess really in higher throughput which genes would lead maybe to fate or state changes in these organoids. And here again, just an exemplary data set where we knocked out transcription factors in developing brain organoids and could see all kinds of composition changes based on the presence of guides and the knockout of the given transcription factors. Here's an example for environmental perturbations. In this case, really in high throughput, performing high throughput screening on organoids. This is a great example from Prisca Liberali's lab. And in this case, what they did, in this case, I must admit, this is mouse intestinal organoids, but of course, this can be done on human organoids. They grew these organoids while treating them with a whole large library of compounds and then performed an image-based screen. And they could identify several major phenotype classes in these organoids, including this regenerative phenotype, where they then could really narrow down what the retinoic acid to be driving this regenerative phenotype and helping regeneration of the intestine. We can use organoids to model disease, and in this case, really of a given human individual, and really perform a personalized analysis. Here an example from us where we generated in collaboration with Silvia Capello from the Max Planck for Psychiatry in Munich, organoids from patients with heterotopia, which is a malformation of cortex development where a subset of neuronal cells fails to migrate to the cortical plate during development. And when we generate organoids from these patients, we clearly see that subsets of neurons are indeed stuck at the ventricle, whereas in control organoids, all neurons manage to migrate in the right way. And of course, now profiling this with single cell technologies allows us to really define and phenotype this altered neuronal state in the tissue. Another great example from Jürgen Knoblich's lab is this application of organoids to study tuberous sclerosis complex. And in this case, comparing patient tissue and patient-derived organoid tissue, they could really see the histopathology recapitulated in the organoid with these subependymal nodules. They could see dysmorphic neurons and giant cells in the organoids. And by performing single-cell RNA sequencing, they could identify populations of cells that were really upregulated in the patient-derived organoids. And this really highlighted that late caudal ganglionic eminence derived interneuron progenitors are really amplified in TSC and lead to this pathology. We can, in the organoids, directly trace lineages. In this case, we developed this method iTracer, where you can now introduce into these Cas9 inducible iPS cells a vector where you have a barcode in front of the GFP, which serves as a lineage mark. And at the same time, you can, at a time point of your choice, scar the GFP. And this scar that is highly diverse can be read out as another heritable lineage mark. And you can generate organoids, pretty much any organoid type from these iTracer iPS cells. And then in addition to phenotyping the cells and really analyzing what cell types are emerging in the organoid system, you can establish lineage trees for the organoids. And of course, this will be particularly interesting when then comparing a control or wild type state with a mutant or diseased state. One can also, of course, directly measure lineage relationships in the cells using live microscopy, in this case, light sheet microscopy. And there are really ways to measure or to image these organoids over many weeks, which allows you to then directly track lineages. And now coming back to this point that we want to profile these engineered cells and tissues. And actually, in this case, we can't just add to the human cell atlas, but the human cell atlas on primary tissues really helps the organoid field, since through the comparison of the reference and organoid atlases, we can really assess quality of our cell and tissue engineering. And when we do this, of course, we might find that, for example, there are cell types in the organoid missing that you find in the primary tissue counterpart, or you might have some off-target cells in the organoids. Also, you might want to compare really differentiation trajectories and find out why, for example, organoid cells fail to mature in the right way and which genes might be associated with a proper maturation of the tissue. And here just two fast examples for how the comparison with primary atlases can then be really used to reverse engineer or better engineer the human tissue. And this is an example that also Mus brought, where, you know, Greg Camp, Jason Spence, and my lab profiled primary organs during development and compared a human intestinal organoid time course to this primary reference atlas. And this allowed us to find out that really some of these organoid cells are of rather foregut origin. The majority, however, is really intestinal cells, as we would expect. But now we can go in and ask what is different between the organoids and the primary atlas. And in this case, we identified a factor NRG1 that can be added to the culture and that really helps further mature and better mature these in vitro iPS-derived tissues. And finally, an example from Lisa Little's lab, where they started to really combine organoids with cellular extrusion bioprinting. And what they could find is that if you print organoids instead of manually generate them, and in particular print them in lines, as you can see here on the right, as compared to in dots, they could see a much reduced amount of unpetaled tissue, which you find here in the middle, and a higher maturation of nephrons, which they could identify by profiling the single-cell transcriptomes of these different types of organoids. And with this, I'm at the end, and I'm very happy to take questions together with Christoph.
Thank you, Christoph and Barbara. That was great. So we have one question, time for one question from Malta Lucan, who asks, how many samples do you need to confidently distinguish between compositional changes from organoid perturbations and random compositional variation between organoids? I guess natural variation.
Yes, I mean, this is a great question. The advantage of these pooled approaches is, of course, that you can have control cells present in the same organoid. And of course, you might wonder, by knocking out certain genes that would be very essential for patterning, you might even perturb the control cells in the organoid. But I think this type of analysis in these pooled approaches really gives you some control over these fluctuations that you see otherwise. Also, I would add that generally you see bigger changes when you use different lines. So there's definitely a line-to-line variability. If you work with the same line, different batches and different organoids are a bit more controlled in the composition.
Great. Thanks, Barbara. So I think we are out of time for our questions, and I'd like to really thank all of the speakers in the session for their really informative and exciting talks. And we're now going to convene our HCA Biological Network discussion panel. So thank you all the panelists for joining us today. We're going to set up the panel. So while we set that up, I just wanted to give an introduction that I'll wait until the panel's set up. Okay, great. So our panel is now virtually convened, and I, again, like to thank all the panelists for joining us today. So the Human Cell Atlas Project Biological Networks represents groups or collaborative networks of scientists who are coming together to, based on shared interest in developing a Human Cell Atlas project in a specific area, such as focusing on one tissue or mapping genetic diversity. And you heard about some of these already today. So importantly, biological networks are open to anyone, and they cross-cut individual projects and grants that people may be involved in. And these networks, I think, form a real heart of the overall Human Cell Atlas project because they're responsible for generating a lot of data, although it's not the only part of the Human Cell Atlas project because there are also working groups and many other activities. But in this panel, we'd like today to discuss how to build a successful and healthy HCA biological network, as well as set the stage for individual biological network breakout sessions that will take place later today. So I was wondering if we could start with introductions. So if each of the panelists could introduce themselves and briefly describe the network that they represent. I guess some of you have already described that in your talks, but not everybody gave a talk. So you can choose how long you want to spend on that. And I'll just start. I'm Gary Bader from the University of Toronto, and I work with the Liver Network. I'm one of the coordinators for the Liver Network, along with Alan Mullen. And I'm also involved in the development and pediatric networks, or I follow that one at least. Okay, Sarah, do you want to go next?
Sure. I'm Sarah Snelling. I'm from the University of Oxford, and I coordinate the musculoskeletal network with 3 other coordinators. We've got quite a big job because the musculoskeletal system composes about 30% of body weight, and we're working across all the different tissues there, from sort of development through to healthy aging.
Great. And we'll follow the speaker order for today. And you don't have to go into a lot of detail if you don't want, if you've already spoken. But Maz, do you want to go next?
Hi everyone, Muz Hanifa. I'm based in Newcastle University and Wellcome Sanger Institute, and I'm one of the co-coordinators of the Developmental Biological Network.
And Martijn?
Okay, hi everyone, I'm Martijn Awijn. I'm one of the coordinators of the Lung Biological Network of the HCA. I'm located in Groningen, the Netherlands, and the other coordinators of the network are Sasha Misharin, who also presented earlier, and Jay Rajagopal from Mass General, and then Pascal Barbry from Nice.
And thanks, Martijn. Christoph?
My name is Christoph Bock. I'm at the Center for Molecular Medicine of the Austrian Academy of Sciences and the Medical University of Vienna. I'm co-coordinating the Organoid Cell Atlas network, biological network, together with Barbara Treutler.
Okay, great. So we'll monitor the Q&A part of the website to take questions from the audience. So please keep those coming in. But we also have a series of topics that we'd like to discuss. So the first question I wanted to ask the panel is, why should we start these biological networks? So what are some of the advantages of forming a biological network compared to just working ourselves and not connecting with other people? And anybody want to take that question? So feel free to use the raise hand feature if you want, or just raise your hand.
I can start, maybe.
Okay, go ahead.
Certainly, I think there's actually a great video clip of like these caterpillars moving together as a group on a tarmac. And you know, when one caterpillar is moving, it goes really slowly, but when the caterpillars move together as a group, they're going really fast, like the tires of like a army vehicle. And it's exactly like that. You have so much more momentum as a network. You have so much more expertise. Your network is suddenly, increased in orders of magnitude because, one person, has contacts with all of these other networks, and then another person, you just bring it all together. I just cannot see how we can achieve anything at scale without bringing in lots of people and working as a network. And certainly for development, Gary, you've been very much part of this, and you're one of the organizers of the Developmental and Pediatric meeting. You've just seen the tremendous progress that we've made over a year in terms of defining what we need to do for the developmental network, what we've achieved, what we lack, and where we need to go.
Great, Maz. And Christoph, did you have something to add?
When the Human Cell Atlas started, it was a kind of comparatively small group of people, but it has grown dramatically over the years. So in a way, these biological networks also provide this kind of small group environment where stuff gets done, and where kind of a team or kind of a small to moderate number of people get together and just move the science forward where there is need for coordination, but at the same time, not with the full complexity of doing something atlas-wide.
Yeah, and one of the things I've always, if you stop and think about what we will need to finish the atlas and how long it will take, how many samples, if you just do a back of the envelope calculation on number of samples, you realize that it's a huge effort. You might need tens of thousands of samples or hundreds of thousands of samples. So, really, there's no one lab that's ever going to be able to do that. So, and if you really want to have an integrated reference atlas, you have to coordinate. So, yeah, great point. Sarah, you wanted to?
Yeah, I think a lot has already been said, but I think so. The musculoskeletal network's very new. And as I said, we're very large in terms of what we're aiming to do. And we're at the stage where we're trying to outline what we need to do as a network. And I think that's where that interdisciplinarity and that sort of mass effort is really important, because there's so many priorities for us in terms of even the tissue we should be looking at. And what we want is an equitable atlas, and that's only going to come through this coordinated biological network activity. And I think probably one thing I'd like to say alongside that is actually you have this focus group within the network, but actually coordinating with the other networks is very important. For example, for us to do development in isolation when there's a development atlas would not be the sensible thing. We need to talk between the networks as well, but having network coordinators adds an efficiency to that scale of communication, perhaps.
Great, and Maz?
I actually want to bring up a point that, you know, we all recognize the value of working as a network, working collectively, collaboratively, but something to bear in mind is essentially how this approach is not necessarily the common research culture that is prevalent. And actually there's a lot of importance in articulating this, what it means to the junior researchers, what it means to how output is recognized, plurality in terms of recognition, what it means for people to share data at a much earlier stage than a lot of other people will be comfortable with. And there needs to be a change in mindset within the researchers. You know, the community must lead this, but we need to talk about this. And I think funders need to also be aligned to actually encourage this, because if there is value in this approach, it needs to be supported.
Really good point. And Martijn?
Yeah, I think that's a great point, Moose. And there's just one example of that is where we had all the COVID papers with the Learn network, and even expanding into other networks within the HCA, where we basically had a group author where all the senior people like myself and all the known people from the field were just in a group author, and all the named authors were actually juniors. That, you know, and then we already had this huge list, but that's in recognition of the fact that they do a lot of the work, but it's also, you know, the possibility that they can really contribute. The other thing I think, which is really, really important for a network like this, is that it's also a way to disseminate these technologies and methods and protocols where otherwise, you know, the advance of these sort of techniques is just much slower. So it's being adopted by all these different labs, and they're all talking to each other. How do you do this? How do you do this? And that improves the methodology in such a way, and that's both wet lab and dry lab part of that. That the fact that you're working together really accelerates the whole process. So that, you know, it's not just that we generate more samples, it's also that we're better at it, quicker at it, and you know, that stuff just gets done efficiently.
Yeah, I'm still thinking about Maz's analogy of the caterpillar, and if I'm a single caterpillar, I want to join this amazing structure that all the other caterpillars have made, but I also want to sort of figure out how that relates to, you know, my life. And I guess one of the ways that we can encourage people to do that is to just reach out and have that discussion, right? If people are concerned about, you know, how they're, you know, especially as junior researchers, how their contributions are tracked, reach out to the biological network coordinators and have that conversation. And actually through that, we might be able to share experiences across the biological network, since it's a general topic. Okay, so I wanted to move on to another topic, which is how you as a biological network coordinator work with the ALICE project to define what the ALICE is, and what are some of the challenges that you're working on overcoming, like how to access tissue and how to prioritize.
I can kick off with our own experience within the developmental network, and in some ways, for us, you know, running the seminar series and writing the roadmap, you know, brought together a lot of information which was subsequently distilled and provided the clarity of where we are, what we've achieved, and where we want to go. So I wonder if these types of, activities and exercises is actually really good to sort of like just, when we started, Gary, there was just so much, like, the roadmap was like, I don't know, 100 pages long, but eventually distilled, and the salient points kind of like coming out of that. So this is a good strategy perhaps that other networks might want to implement.
So maybe you can talk more about that, how, what is a roadmap and why is it important? You mentioned sort of how to create a roadmap. You bring a lot of people together and write a big document and distill it, but what is the, what's the value of the roadmap?
The value of the roadmap is to kind of prioritize what are the next steps and what are the types of projects we need, and so we can be more concerted and focused with where we want to go and how we can actually, generate the funding that is required for what we need to do. Bring in the kind of communities, reach out to the communities that we need, to link up with. And I think in our case, it was very clear that we really do need to work together with the pediatric community. And I think, pediatric cell atlas merging with the developmental cell atlas is going to be absolutely transformational, particularly with disease data sets in the pediatric setting. Those kind of realization. A question Dana asked before, the computational challenges, is only when you do these works or plan to do the work that you anticipate some of the missing components that you really need, and you can start building them. How do we actually provide this atlas? What is going to be the tangible output from the atlas that people can actually look at? We're all very visual as human beings, What do we want to see at the end of the day? All of those things are the kind of like concepts that came out from our discussion.
Okay, great. Thanks. And Sarah, did you want to go next?
Yeah, sure. So I guess on the challenges, we're a very new atlas. And I think for us, it's actually establishing quorum in terms of our membership right now, because what we need is that, we need that group of caterpillars effectively, and we need that to be an interdisciplinary group. And I think, as Muz kind of alluded to earlier, it's getting through the idea that this is a collaborative network of people. It's not about funding at this point. It's about working together to make everybody more efficient and to be transformative and to push progress. And I think for us, it's engaging with our community to make sure that the researchers understand that and want to be part of that. They see the benefits for them. And beyond that, our challenges as an atlas are prioritizing our tissues, which is why we want quorum, because we've got a large musculoskeletal system that's affected by a number of diseases across lifetime. So which tissue should we be looking at is one of the major challenges for us in developing our roadmap. And how do we access those tissues is another big one for us. You can't easily get them. It's through biopsy, it's through surgical operations, waste tissue. And that's where the network can be beneficial, because the experience of one team can guide another team in how to access these tissues in terms of the ethics, in terms of surgical sources. And I think finally for us in terms of the challenge, it's in general, the musculoskeletal system consists of tissues that are cell poor and extracellular matrix rich. And this is again where this network benefit comes off, because people are working to develop protocols for these types of tissues. So the amount of time, the amount of money, the amount of effort that can be saved through that protocol sharing is really huge. And that's something that we're really trying to do. We've developed some single nuclei methods that we'd love the community to use, because if that can get data out there earlier for everybody, the progress towards disease atlases, to the atlases across development and lifetime is going to be massively expedited.
Great. And Martin?
Yeah, I just, responding to Sarah and Moose's points, but also to what's being pointed out in the chat or in the questions, there's this issue about trust, basically, because if you start to work together as a network and you agree to share data and methods early on before publication, it requires a certain basis of trust that you still allow each other also your individual publications, because you can't say, everybody just throw the data on one big pile and we'll make one big publication, which in our case would be the lung cell atlas, and that's it. because people need their individual outputs, they need their individual, have the deliverables for the project, they need visibility, etc. So within Lung, this really started as a number of parallel groups working, you know, in parallel on their own islands, if you wish. And then we really valued the, you know, the added value of working together, sharing early and coordinating, but still allowing everybody to have their own publications. And, you know, there's no need to get in the way. So we never had something like a memorandum of understanding or anything else. We just try to coordinate as much as possible where we could, but still allowing the individual members to also be productive in their own right. And that, I think, worked so far very well. And you can also see in the field, for Lung at least, that there is like parallel developments happening in multiple places. And they actually make each other stronger, because then you say, okay, so if you take this exact approach, what is that really good for? And, you know, you take a slightly different approach and you can answer specific questions better, but other questions not so good. So you also learn from the breadth of the network. And so I think what I'm trying to say is that this trust and this working together really pays off in the end. As long as you know, also allow people their own independence, because otherwise nobody would want to contribute, I think.
Great. And Maz, you want to add to that?
Yeah, I just wanted to add two things. And within HCA, there is the data sharing principles according to the Fort Lauderdale principle. And I think that is actually very helpful. It means that, you know, you can share data early and having that sort of protection that Martijn alluded to with regards to, you know, a group just doesn't take someone's data and publish it in its entirety and sort of like, you know, leaving the junior researchers in the lurch, so to speak. So I think those type of guidelines are actually extremely important to sort of talk about and actually disseminate and adhere to. The second thing I think I want to mention is Martijn also alluded to the idea that, you know, with the COVID pandemic, and in many ways, the type of work that we were doing for the Human Cell Atlas really gave us pandemic preparedness. So when the pandemic came, we were almost ready as a community with the kind of like technologies we were using, with the type of, you know, research framework that we were undertaking, the team science, collaborative. It just seemed like, you know, you can just pivot very easily. And, you know, if you really think about it, it wouldn't have been so easy to pivot. And, you know, as Aviv and Sarah mentioned yesterday, I think there were about 17 publications just within the last 18 months from the HCA community towards COVID-19.
Yeah, you really require a network to be established for that to occur, because otherwise you don't know who to speak to. There's an interesting question from Gloria Freihuber, who kind of brings a couple of things together that we've been discussing. So, you know, she mentions that a concern people may have about sharing data early is that it will affect their ability to secure funding, and she's mentioning that funders need to figure out how to offset this concern. I wanted to mention that, you know, when we write this roadmap and organize together as a network, we actually provide the ability to connect with funders who would much prefer, presumably, to work with a network of coordinated individuals rather than lots of individual people, and also to have that network prioritize their work by writing a roadmap and going through that process. That really helps define the challenges and that the field agrees on, and then you know, identifies funding opportunities to address those challenges. And I suppose that if you're in the right position by contributing to all of those, then you're ready for those, and you also have a connection with the funders. So maybe that's one way that, you know, we can think about this more broadly, that the network doesn't, it's not just scientists, it's multiple different people, not just, you know, people working in the lab on Atlas efforts, but we can think about the network being a broader connection. Christoph, you wanted to add to that or make a point? You're muted, Christoph. Please unmute.
Ultimately, the science is done by people. So, and trust is also among people. So yes, we can make structures, we can make memorandum of understanding, but we need to make sure that people meet each other. and meeting we have shown now over a year can happen in the virtual room, but in a way the biological networks create this framework in which people meet each other, work together, can toss around ideas, and from there kind of bottom up is growing the trust to do things together in the same way as or complementary to the Human Cell Atlas, kind of top down, providing a shared set of values that people kind of subscribe to when getting involved in the Human Cell Atlas. So while I think that kind of values is providing the roof and the framework, it's really this kind of personal connections that the biological networks can provide that builds the trust from the bottom up. And in this way, I think it's also very important to emphasize in this context that the biological networks are really open to new people getting engaged. So this is by no means a closed club. This is something very much in flux and very much kind of work in progress that is entirely open to new ideas, new perspectives, new people.
Great point. I wanted to take a question from Bruce Arno, who asked about genetic privacy. How do we enable rich analyses without compromising donor protection, in particular for children? But I also wanted to just expand that to a more general topic that is about equity, diversity, and inclusion. So, you know, how do we support all of these things that we might not always be thinking about when we're, you know, thinking about how to get our NucSeq protocol working, and what does that mean? So, Martijn?
Yeah, I think the success of the network lies not with the coordinators in a sense. So it lies with its members. So as the lung community, we've been very successful in getting cells from white males, middle-aged preferably. We have lots of those. Other ethnicities, age ranges, they're heavily undersampled. So this also reflects where the early initiative was, in which locations, and focus on what sort of ethnicities, and we clearly need to improve on that. That's very urgent, but that also requires the network to broaden. And I can't just start a lab somewhere else. People that are in other places, please contact us and join us and start to contribute. And that is really, 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 we'll really, really appreciate this. So please do and help us here.
Great, Maz?
I wanted to add to that point. I mean, I think it's very important to be diverse and inclusive, and definitely, the Human Cell Atlas has to embrace that in its entirety. But there are some issues, which is probably most relevant to the developmental network, whereby you have cultural and societal sensitivities that needs to be considered, and also the availabilities or existence of bioresources that can actually, allow this to happen independently of the researcher's interest and how it interfaces with the clinical, environment. And I mentioned earlier on about, resources like the Human Developmental Biology Resource and, the aspirations and the newly formed Hugh Decker. So those are things that probably we ought to also encourage the existence and their growth to support types of research that will also allow us to embrace diversity in terms of the profiling that we do around the world.
Any other comments about that? I think that, we might need to play some kind of more active role. If it's not happening naturally, we have to, somehow be more active. So I wonder how we would actually do that. Sarah, I don't know if you were answering that question. But
just kind of adding to what's been said as well, I think one of the things that we need to think about with diversity as well is it's not just in our donors, it's also in our teams. And one of the things that we've been very aware of with musculoskeletal, I suppose, is that if we want ancestral diversity, for example, in our tissues, we can't work just within the UK and the USA. We need to reach out to other countries and work in other countries, but that involves us having diversity in the teams we work with. So we need teams on the ground analysing and computationally trained to work on these tissues internationally. And that's something that's, I think, it's not a biological network effort, that's a sort of HCA effort. More generally, you know, we've spoken to teams, for example, in Zimbabwe, and talked about setting up computational training there. And then we also face challenges in other countries, and this maybe relates to the point of the Developmental Atlas as well, is there's cultural sensitivities because of tissue donation, but also in some places, the tissues we're working on, the orthopedic operations that we'd normally source these tissues from simply don't exist. So it's going to be quite, it feels like it's a getting the ball rolling process for a lot of this. Like, we need to start and we need to make it a priority, but I think we also need to be aware that for like Musculoskeletal Atlas 1.0, our diversity is probably going to come quite a lot from teams within countries that are already set up. But within the goals of us as 1.0, we should be establishing sites more internationally.
Yeah, great point. So, you know, in about an hour or so, we're going to break off into our own, you know, breakout sessions for each atlas separately. And I wonder how, you know, and we're actually going to be discussing these same topics across these different sections with different people. So how do we all bring all of that together? And also, you know, with other projects that we're part of, HubMap and other big initiatives that exist, presumably some of these things are going to be discussed in multiple places. Christoph?
I think the advantage of the Human Cell Atlas and the biological networks in particular is that they are not a top-down structure in the sense that you can only be part of it if you have funding through the Human Cell Atlas. It is in this way, it has the Human Cell Atlas has been able to provide a bit of a framework for initiatives from various funders, also from national and regional funders, to work together in a meaningful sense. And that is clearly the case for the lung, as Martin has already explained to us in detail. But more broadly, because the Human Cell Atlas does not have kind of a very strong NIH branding or EU branding or whatever type of funders branding on it, it has perhaps is an easier position to provide some kind of a clearinghouse for standards and interactions. It's also less dependent in a funding agency losing interest or moving on to other things, because it's to a significant degree driven by the people who are actually doing it.
Any other points? We're almost out of time. Yeah, I mean, it's important for everyone to understand that, you know, my personal view is that the HCA is a global effort. It's an idea by Homo sapiens to map Homo sapiens, right? So however you're mapping, you know, you're really contributing, and it doesn't matter if you're part of different projects. So, but we do need to think about how to coordinate all of that, and it might be an interesting topic for the breakout sessions later today. Just before we end, what maybe each person could say something about either advertising their own breakout session or explaining what you, or mentioning what's going to be a really interesting topic that you would love to talk about at the breakout sessions that are coming up. Anyone want to go first?
Me too. Oh, so
Sarah, sure.
The musculoskeletal network, which we would love members for, and I think this is where actually, if you've got an interest in musculoskeletal, please do come along, because I think we can learn a lot from other teams, for example, working on extracellular matrix-rich tissues, and I think we can also inform teams working on development as well. And even with organoids, we've got models there. And I think one of the topics we'll be actually talking about, because we're very new, is priority tissues, and how do we prioritize the network focus on the tissues there to sort of deliver benefit towards the disease side of this in the long term.
From the perspective of development, I think you know, how do we, the next phase will be how do we actually interface with all of the networks that are around, because, you know, we cover so many organs, but they're just a different age range. And therefore it's highly relevant to all of the networks, and forming this sort of like seamless interface so that, you know, everyone can actually benefit from the data, getting the most out of it, and you know, disseminating this. And if we can do it within the sort of like community of networks, and then that will give us a template of how to kind of interface with the broader, with the other networks on, you know, other consortia and initiatives that are ongoing. That I think would be an interesting discussion point.
Okay, so very quick things if anyone wants to mention, because we're running out of time. So
I'll just do a very brief shout out for Lung then. So we really want to discuss, you know, what's the next step within the Atlas, both, you know, all this diversity aspects, but also very concrete, how do we harmonize cell type labels? How do we integrate it with ontologies? Because there's a lot of noise, if you wish, in literature. And we feel that we should just get together and get organized on those aspects so that as a, you know, as a network, we can really move forward.
So in the Human Cell Atlas, so much cool new biology is discovered. This must have the potential for many, many new drugs to be developed. And I would think that our organoids are the platform in which we should do a lot of drug development going forward.
Okay, great. So, yeah, I mean, maybe one of the things that we can think about at this breakout session today is also topics that we should highlight that other, like the ones that you just mentioned, that the other networks should know about, right? So it's kind of like writing the title and abstract and intro or something for your paper. You summarize it in a short way, and then that's what's easy for other people to sort of quickly understand. So I wonder if we should do that for all the networks, and we could think about the topics that, so that's kind of maybe the more unique summary aspects, but also the cross-cutting themes we can discuss at these breakout sessions. Okay. Thank you very much, everybody, for participating today. We have to move on, and we will see everyone at the breakouts. Thank you very much.